Hello learners, welcome to NIOS Senior Secondary Biology course. I am Dr. Babita Trangukola, Associate Professor, Department of Botany, Zakir Hussain Delhi College, University of Delhi. Today we shall be discussing the topic related to growth and development in plants. Now this topic is divided into two parts, and the objectives of part 1 are to discuss and define the difference between growth and development, to explain the growth curve and the various stages involved with the cellular growth. Also to know the various methods how to measure the growth of plants and describe the factors that impact plant growth, and also most importantly to discuss the importance of growth regulators. Now this will also involve an activity in which we shall be using a direct method to measure the length of root and shoots by simply taking the plant and measuring its length with the help of a thread, and that shall be measured on a meter scale so that you get an approximate idea about the length of various parts of the plant. Now what is growth? Growth is basically the most important, fundamental and conspicuous character of living beings, and it is defined as an irreversible increase in the number and size of cells, organs and of the whole organism over a period of time. Now, what is the development? Development is basically composed of two important parameters, which are qualitative and quantitative changes that a plant grows through, such as growth, differentiation and maturation, which an organism undergoes throughout its life cycle. So, which means the development of a seed into a mature plant is a very interesting example of the case of course, both this growth and development. And they are two parameters which have their control in the genetics, in the genes of the plant, which means if a plant has a particular trait, say you can see here shape of the leaf or structure of the various roots, above ground parts and below underground parts, all these traits, whether it is shape of the leaf, color of the flower or size of the fruit or any particular trait. There are so many traits that you can see plants have, and you can actually take a plant in your hand and see, and you can enlist many characters, minimum 40-50 characters can easily be identified. And the important thing is that these all traits, whatever we think maybe as a very simple trait, say apex of the leaf, this is also governed by a particular system, that is the genes which are present in the plants. Anything that you see, it could be margin of the leaf, which can be smooth or it can be serrated. Now these are two different traits, so they are also governed by, these are two different forms of a trait, they are also governed by the genes. So which means if a trait is missing in a plant, that means either that gene is faulty or that gene is absent. So they are to tell you that these two processes have their control in the genes of the plant. Now in this particular slide, you will see growth can be measured. On the left hand side of the figure, you will see there is leaf which is smaller in size, and you can see this leaf which is marked as A, and you can see the leaf on the right side which is marked as B. Now both the leaves over a period of time have increased in their area, and you can see the demarcations as A1 and B1, which means both the leaves have grown in the area. That means growth has taken place undoubtedly. However, an interesting thing that I would like you to note here is that here it is relatively, although the size of the leaf is small here, but relatively more growth has taken place in the small leaf compared to the larger leaf which is on the right hand side. And also we can see on the figure, on the right hand side, where you can see a seed is there. The first figure in this picture on the right hand side, you can see this seed is there, and there are various parts, that is seed coat, and this is hypocotyl, and these are roots. When it undergoes development, now you can see slowly how different parts are elongated and how different parts are being added up. Here more parts are being added, you can see epicotyl, and the root system has also become much larger. Eventually from this seed what you are getting is a complete plantlet. Now see the number of leaves that the plant has produced. Now, the transformation of this seed into the plant, where each organ is clearly differentiated into root and shoot and leaves, this is what is development, and this is a case of bean that has been discussed for your understanding. Now, coming to growth and development, to make it little more clear for your understanding, growth in living organisms is not uniform throughout their life cycle. Initially, the growth is very fast till the plants or the animals, they attain maturity, and then it slows down and ultimately it stops. Now, in the case of development, it essentially involves two important parameters, that is, development is made up of, is essentially comprised of morphogenesis and differentiation. Now, what is morphogenesis? Morphogenesis is the look, the structure that you see in a plant, which means the process that leads to the development of shape and formation of the structure of any organism. So, morphogenesis basically means the look that you see, the morphology that you see. That process is known as morphogenesis, and differentiation is when the cells change in the process of maturation and they take different roles. And you know, the cells which comprise of several cells which comprise one tissue, they are all alike, and then these tissues can combine together form organs, and which carry out very essential important functions of the entire system to sustain life. So, I hope it is clear that growth is something that is irreversible, and it can be measured for different organisms in different ways, and development is essentially comprised of morphogenesis and differentiation. Now, coming to stages in cellular growth, there are mainly three main stages that are present in the cellular growth, that is cell division, cell enlargement and cell differentiation. Cell division is mainly possible due to mitosis, and cell enlargement is the increase in the volume of the cell because of the increase in the volume of protoplasm. And differentiation, as we have just mentioned, that differentiation is when the cells during the course of their maturation become so specialized that they can perform a particular function. Now, these living systems, they follow a particular pattern during their growth, and we have seen that if we try to plot a graph between two parameters, this graph essentially here shows a plot between two parameters, which is on the y axis. You have on the y axis here, you can see this is the growth parameter, which can be measured in terms of size or weight of the organ, and on the x axis you have time as a parameter. And initially, as you can see here, when the growth is in the initial phase, it is very, very slow, and this slow phase is known as lag phase, and soon it picks up and it grows exponentially at a very fast rate, and during this period the cells have the potential to divide, and they continue to divide till nutrients become a limiting factor. Till the time nutrients are available in abundance, they go on increasing, their growth goes on increasing, and this phase is known as exponential phase or log phase. Now, because the nutrients have become limitation and the growth slows down, so therefore the growth curve becomes stationary. So, this phase is the one where the growth has dropped. Now, how can we measure growth? The measurement of growth can be done in various forms, and there are different ways to measure growth. In the case of stem and root, it can be measured simply by noticing down the increase in the length of that particular organ. In the case of leaf, as you have seen earlier, it can be measured in the terms of increase in the volume of that particular leaf, and in the case of fruits, it can be measured in terms of leaf it is area, and in terms of fruit it is volume that is measured to measure the growth of that particular organ. However, in the case of some algae and yeast and especially bacteria, the growth cannot be measured in terms of length or volume. It is measured in terms of increase in the number of cells. Now, the growth can be measured directly, as I am going to show you an activity here. For your convenience, you will see there is this plant, and this part of the plant, which is the shoot, and at the bottom you will see these white structures which are the roots. Now, it is very simple and easy to understand that we can measure this growth. This is a straight shoot, so it is easy to measure the growth, but nonetheless I will show you a method which can give you an approximate idea about, if not an exact idea, but an approximate idea about the growth, especially in plants which are growing in the field and which do not have a straight shoot. We use a thread. It is a simple exercise that you can perform. We use a thread and mark the length right from the tip to the bottom of the shoot. Yes, I will show it from this side so that it is clear to you, and I will mark this. I will hold the other end and we can straight away measure it on a scale, meter scale, and here I can see that it is 20 centimeters long. So, I can note down that the shoot, if I am trying to show you the growth, this would be growth is always with respect to time. So, this would be in case I am trying to show you this for a particular time period, and this is an experiment where we are trying to measure height over a period of time, then we will see initially the plants will be small, as you can see here. In this there are small plantlets which have taken much lesser time compared to this one which has taken, bamboos are essentially they take time to grow. So, this is essentially a bamboo shoot that has taken quite a bit of time compared to the one which you have seen just now, where the length of the shoot is lesser. Similarly, it is an easy method and a accurate method, almost an accurate method, we can say that these white structures are there. Sure, you can see these white structures here, and you can take again this thread and hold it against the root and make the root straight, so that you can measure the length right up to the tip of the root, because the root involves the entire length from the point where it emerges from the bottom of the shoot till the tip of the root. This part I will again take meter rod and measure it. So, this shows it is 4. So, one thing I can clearly show it to you, that the length of the shoot is much larger than the length of the root. Now, is that the case with all the plants? No, you can try at your home, take onions, put them in water and keep noticing from the side from which the roots come out, and keep noticing every day, and you will see over a period of time many roots will grow much longer and some will not grow that long. And also I have got another plant which is garlic. Here you can see, as I was telling you, the length of the shoot is much larger. This is another case where you can see garlic that has taken 7 days, because it is cold these days, and it has taken 1 week to reach up to this stage. Can you see the roots clearly here, white structures which you have earlier seen here, and this is the shoot part. Now, if I have to take the length of this, I can straight away use this over the meter rod and note down the length of the shoot. Likewise, I can do it for couple of samples, and then I can take average length of the shoot in a period of say 1 week in winters when the temperature was around 15 to 18 degrees centigrade. How much was increase in the length of root and shoot? I can do the same experiment in different periods, may be in summer, and see is there any increase when the temperature increases, and you can understand very well that yeah, there are various factors which play a role, critical role in controlling the length of the root as well as shoot. Then there is yet another accurate method of measuring growth of plants, which is by Auxanometer. What we did just now is of course a very approximate measurement of the length, but however, if you want to have an accurate measurement of the length, and you can use Auxanometer. It is a simple equipment which is designed in such a manner that it can give accurate measurement of the length of the shoot. Now, in this what happens, there is a potted plant. This can be performed in a potted plant, and in this the tip of the shoot is tied to the tip of the shoot is tied a thread, and here yes, you can see this thread is tied to the tip of the shoot that grows in a pot, and the other end goes over the pulley, and a weight is attached to the other open end of the thread. Now, to this pulley there is also a needle which is attached, and this needle slides onto the scale of this arc. There is this arc and you can see the scale. So, this needle slides on this arc. Now, what happens when the plant grows? Because of the weight, the thread will come down and the needle will show deflection. Depending on how fast or how slow the plant is growing, it will show accordingly the deflection on this scale. So, we can measure these from the scale, we can take the reading and see in how much time, how much of needle has deflected, and we can take a measurement of the accurate reading of growth of plants. Now, we will talk about the factors that affect the growth of plants. Now, there are several factors which affect the growth of plants. Now, first of all, we will talk about the external factors, in which one of the most important parameters is light. Now, besides the essential role that light has in photosynthesis, light also plays a very critical role in the germination of some plants. Although light is not required for the germination of most of the plants, because seeds are grown inside the soil. However, in certain plants where the seeds are tiny, for example, snapdragons or seeds of Nicotiana, that is your tobacco, the seeds are small and they are sprinkled on the surface of the soil and they require light for their germination. Light is also essential for the growth of seedling, for the differentiation of tissues into various organs, and also for the reproduction of the plant. Another parameter that has a crucial role on the growth of plants is temperature. Now, we all know that temperature plays a crucial role on controlling the biochemical activities of the plant. So, there is an optimum temperature range in which most of the plants are very happy and they grow healthily, that is between 28 to 30. However, plants can still manage to grow in a temperature range from say 4 or 5 to 40 and beyond 40. They can sustain that much of variation also in the temperature. And if we subject plants to very low temperatures, then they are going to suffer due to chilling and freezing. They are going to suffer, and that will cause internal injuries to the plant. And of course, very high temperatures will tamper with the enzymatic activities of the plants and they will stop growing. Yet another parameter, external parameter that has a role in controlling the growth of plants is water. We all know water is an essential commodity, and in plants have in their entire system more water than animals need. Almost more than 80-85% of water is needed by the plants, and these plants have an essential role in all the biochemical activities, and in addition they have a crucial role in maintaining the temperature of the plant and also in supplying nutrients to the plant. Now, water is essential undoubtedly, but water deficiency or excess of water is also detrimental for the growth of the plants. Coming to another parameter, that is minerals. We all know mineral nutrition is extremely important for the growth of the plants. Now, depending on whether minerals are required in a particular amount or in a low amount, they are classified into major nutrients and minor nutrients. The nutrients that are required in more quantity, they are known as major nutrients, which are like your carbon, hydrogen, oxygen, and your NPK, that is nitrogen, phosphorus, potassium, and minor nutrients like boron, iron, magnesium, they are required in much smaller quantities. Now, whether they are required in much smaller quantities or they are required in more quantities, they are essential for the growth of the plant, and if they are absent due to any reason, then it is adversely going to affect the plant and it will lead to the loss of crops, and it can adversely affect the photosynthetic ability of the plant. So, all of them, whether they are required in more quantities or in less quantities, they are very much required for the growth of the plants. Now, coming to yet another part of the factors, which are the internal factors, they play again a very crucial role on the growth of the plants. These are the substances which are produced by the plants, and they are produced in one part of the plant, and then they can move to the other parts where they execute their role, and these are known as hormones, and especially in the case of plants they are known as phytohormones. So, phytohormone is a substance that is produced in one part of the plant in a much smaller quantity, and then it moves to the other parts of the plants. It executes its assigned role. There are 5 categories of phytohormones that are found in plants. So, these are auxins, gibberellins, cytokinins, ethylene and abscisic acid. Now, auxin, there is a Greek word aux, which means increase in size, enlarge. So, the main basic role of auxins is to increase the cell size and of the stems and coleoptiles. And it has been seen that auxins are produced in the apical bud of the plant. There are auxins which exist naturally in the plant, for which the case is IAA, that is indole acetic acid, and they are also synthesized synthetically, that is IBA, that is indole butyric acid, or 2,4-dichlorophenoxyacetic acid, or NAA, that is naphthalene acetic acid. These are synthetic auxins. The functions, as I told you, the name itself is an indicator that it helps in cell elongation, and there are plants whose growth is under the control of the apical bud, and in the apex, as I told you, auxins are produced in the apex apical bud of the plant. And these plants where the growth is under the control of the apex, these plants do not, this apex where the growth is under its control, they do not allow the lateral buds to develop, and this phenomena, this part is known as apical dominance. And if you have to have lateral growth of the buds, then we have to prune the tips. So, we remove the tips, which means we are removing the supply of auxins, and once the auxins, the tip is removed, the tip is pruned, then the lateral branches start developing and the plant becomes a healthy plant. And it has many other roles also, as you can see, helps in the, it delays the abscission of leaves, and NAA, which is a synthetic auxin, it prevents fruit drop, and interestingly 2,4-D, which is a synthetic auxin, it is also used as a weedicide. Now, this picture is going to clearly depict to you the role that auxin has in cell elongation. See, a seedling has been taken and it is exposed to light on one side. Now, as you can clearly see here, this is a seedling and this one side is receiving light. So, light rays are coming from here and this side is exposed to light. Now, on the opposite side, what is happening here is, you can see in the enlargement, when the part where the seedling is exposed to light, on the opposite side of that, all the auxins from this side have moved to the opposite side, which is shaded side. Therefore, the concentration of auxins here is much more compared to the side where light is with the side which is exposed to light. And because of the higher concentration of auxins on this side, the size of the cells, you can see, has elongated compared to this side, and because of which the tip, you can see, is moving towards the light. So, this also explains you phototropism in case of plants. So, this movement, this bending towards the light is because of the auxins that have produced, that have increased the size of the cells on the opposite side. Now, coming to next phytohormone, that is gibberellins. It was way back in 1920 that there was a Japanese farmer who was working on a disease which had infested the young seedlings of rice. And in this case, what was happening, that these seedlings were becoming slender and tall, and they were becoming spindly, and soon they would fall and the plant would die. And with their efforts, they found that this rice plant was basically infected by a fungus known as Gibberella fugiculae, and it was seen that the compound that was released into the plant, that was named as gibberellin. So, initially it was thought as pest which was creating causing disease and damage to the plants, but later on it was found that gibberellin was present in many other forms naturally in the plant. So, that means it has other roles also in the plant. Then what was happening in the case of rice? Basically, in the case of rice, where it was producing a disease, the quantity of gibberellin was much more than was required for performing any particular activity in the plant. It was the gibberellin of the plant as well as the gibberellin that that particular fungus was producing there. So, the quantity, the concentration of gibberellin that plant was much higher, which was causing abnormal increase in its height, and therefore, the plants would die. So, the other roles that the gibberellin has, of course, you have understood that it helps in the elongation of the stem, and plants are treated with gibberellins to increase their height. It also breaks dormancy of seeds and buds, and it also induces parthenocarphy. So, you can have seedless fruits by the stimulus that gibberellins provide in fruits. Now, this slide shows how gibberellin plays an essential important role in elongation, stem elongation. On the left hand side, see, this is a plant Gaillardia. It is present in on the left hand side, it is present in a rosette form. This is the rosette form, which means cluster of leaves is present close to the ground, and soon when it was treated with gibberellin, this bolting occurred, and you can see beautiful flower that is being produced. Now, normally in plants, bolting occurs for flowering. However, in certain cases we can induce bolting by giving the treatment of gibberellins, so that flowering can be achieved much before than the plant will normally take in nature to bolt and produce flowers. Yet another phytohormone, that is cytokinin. Now, as the name indicates, cytokinin, which means it is a hormone that helps in the cytokinesis, that is cell division, and it is extracted from coconut milk, and it is also present in many other parts of the plant. And this cytokinin, it stimulates cell division, cell enlargement, and cell differentiation. It also prevents aging of the plant parts, and it also inhibits apical dominance and promotes the lateral bud growth into the branches. Now, cytokinins play a role in delaying senescence. Now, here you can see in this picture there are two plants. On the left hand side is a genetically modified plant which is capable of producing cytokinin till particular age and beyond that also. And on the right hand side, it is a naturally normal plant which has lost its capability of producing cytokinin. Both the plants are of the same age. However, the one that is genetically modified, in which the cytokinin is being synthesized because it is modified, is able to retain a lot of foliage, and the one on the right hand side has lost the capability, and you can see a lot of senescence in this plant. Now, this molecular technique has been exploited by the scientists to delay foliage, falling of the foliage, and it can be used to maintain plants in the green state for a pretty long time compared to the normal plants where the cytokinins are not able to function after a particular age. Yet another phytohormone is ethylene. It is a gaseous hormone, and it is found in the ripened fruits. Now, in this case, whenever we see, there is a smell that ripened fruits normally produce. So, that smell, which is very typical, and it is always common, a similar smell that we always, we are able to feel that smell, and it gives a sense that the fruits have ripened. That smell that we are able to correlate with the ripening of fruits is because of the ethylene, and it also promotes senescence and abscission of leaves and flowers, and it also increases width of the cells. Now, ethylene has much more to do commercially. These are tomatoes that have been picked up in the green state, and they were transported, and on the tomatoes which are on the right hand side, they have been treated with 100 ppm of ethylene for 3 days. Although both were collected at the same time and both were collected in the same state, but you can see a change in the color on the right hand side of the basket, where the tomatoes have ripened and they are exported to the groceries once the fruits have ripened. And this particular property of ethylene has been commercially exploited quite a bit, and many fruits like bananas, apples, and so many vegetables, they are being exposed to ethylene to promote early maturity, to promote ripening, so that they can be sold commercially. Yet another hormone that affects plant growth is abscisic acid. Now, as the name suggests, abscisic acid, The name does not qualify for its function, which means it does not have a role in abscission. Although the name is abscisic acid, but it does not have a role in abscission. However, it has a role to play when the plants face stress, generally water stress. So, we know that abscisic acid has many other roles, like you can see it induces dormancy in buds and seeds, promotes senescence and inhibits seed germination, and it also causes closing of stomata. Now, when there is this onset of winter, it naturally brings a stress to the plants, and seeds which are as it is the mature seeds, they have very limited amount of moisture in them, and then it can be easily correlated with the increase in the concentration of abscisic acid in seeds, which means presence, as long as abscisic acid is present in the seeds, they will not germinate. However, if they have to germinate, the abscisic acid has to be washed away. Now, here in this case, which is an interesting case, you will see that there is this maize cob, and in this cob there are certain kernels here, as the arrow indicates, here as well as here. Now, this is a mutant cob of corn, and in this case these seeds, they are not able to synthesize abscisic acid, and you can see here that these seeds, the arrow is clearly indicating that these seeds have started germinating, and the roots can be seen. They have started germinating while they are still attached to the cob, because they lack the capability to synthesize abscisic acid. So, this clearly shows that they play a very crucial role in maintaining dormancy of seeds to probably overcome an unavoidable circumstance, or avoid unfavorable environment. The applications of phytohormones are many. Auxins and gibberellins, they are used to produce seedless varieties of fruits, such as seedless grapes, seedless papayas, and to name, there are lots and lots. We all know that sometimes when we are enjoying our fruits, seed is one part which slightly makes it distasteful. So, there is a great demand, commercial demand for seedless fruits. So, phytohormones are very, very crucial in the application of the production of these seedless fruits. They also help in inducing early flowering. So, sometimes when we have plants which flower, there are plants which are annuals, biannuals and perennials, and there are plants which can be made to flower in the same period. It may be a biannual, that means in the first year it will not flower, it will flower only in the second year. However, with the application of growth regulators, it can be made to flower in the first year itself. That means we are able to achieve would normally not be possible in the nature. And as we have just discussed, they help in the ripening of fruits, which is this property of hormones has been commercially exploited to a considerable extent. Auxins also induce seed germination, and some of the phytohormones, they are growth inhibitors. They stop germination, and they are used in the crops like potatoes and onions when they are stored for long-term storage. They are given these treatments so that they do not germinate, because after germination the quality of the crops would fall. To maintain the quality, they are given the treatment of phytohormones. This is another important aspect of growth and development. Now, we have understood till now what growth is, what development is, what are the roles, internal, external, fine. Basically, it is the cell, the unique and the important ability of the cell, that it is able to perform such unthinkable functions, and differentiation is one of them. Now, what is it? Differentiation is a change in the size and biochemistry of the cell to perform a particular function, and it can be cells, tissues and organs, which when they differentiate, they are assigned by the system, by the plant body, they are assigned certain different functions, and soon after the cells are formed, they differentiate and they are assigned their own roles and they perform their own functions. As an example, we can easily understand that we have vascular tissue in the plant, and it is composed of xylem and phloem. And where from are they developing? They are developing from the meristematic cells, which means meristematic cells have later differentiated into xylem and phloem. And of course, we know xylem and phloem, they have very different roles to play in the plant. Similarly, there are other examples where the cells differentiate and they soon take on their roles, and overall they work in association with each other and do certain unthinkable tasks that normally one wouldn't think, maybe lifting of water from the soil to so many meters tall, to such heights in the plants which normally one wouldn't think. What is the force that is working there? Yes, it is the special structure of the xylem vessels that is helping in the upliftment of water. So, this is where cells are differentiated to perform a particular important function. Then while we know that the cells are differentiated to perform their roles, and meanwhile they have also lost the capacity to divide and further and do any such roles. However, there are certain cells which regain the capacity to divide and take certain important roles. For example, interfascicular cambium, cork cambium, they are developed from parenchyma cells, which means parenchyma cells were, as it is, differentiated cells which were performing their own function, but now because of dedifferentiation these parenchyma cells are able to produce interfascicular cambium and cork cambium. So, that means a differentiated cell is regaining the capacity to perform under different conditions. Redifferentiation. So, while the dedifferentiated differentiation is going on, if the cell is not able to do so, it loses its capacity to do so. So, it is redifferentiation. A unique property that plants have is, they are very smart. Depending on the environment that they face, they can actually modulate their own structures. So, they have this plasticity, they have this ability to modulate their structures depending on the environmental conditions. So, a very interesting example is heterophyly in cotton, coriander, where you will see there is a difference in the juvenile leaves and in the leaves produced by the mature plant. Or we can also see a plant which can grow on land as well as on the aquatic system. There will be definitely difference in the structure of the plant, of the leaf when it grows on the land, a terrestrial ecosystem, or in the aquatic ecosystem. So, this is a very unique ability, the smartness of the plants to adapt according to the environment that they are facing. With that, we come to the end of the first part of the talk. Thank you.